speaking of California, for our third story, we're now going to move into the immunology space. Headline: Hidden viruses in our DNA could be medicine's next big breakthrough. Scientists at La Jolla Institute for Immunology, when they're not out surfing on the beautiful sand beaches of San Diego, have decoded the 3D structure of an ancient viral protein hidden within our DNA.
Our own DNA.
Our own DNA. This protein found on cancer and autoimmune cells has a unique shape that could unlock new diagnostics and therapies. This was published in Science Advances. Again, this is another California university. We have the most papers, the most patents, the most Nobel laureates. I just always have to make sure people know that we are winning.
Yeah, we are winning. We are winning. This is La Jolla, California, baby.
Yes.
It's right next to UC San Diego.
Yes.
UC San Diego is an amazing crucible for biotechnology and molecular biology research. It's kind of like the Silicon Valley for biology in some sense. And this is a really groundbreaking achievement. It's the first time that we have ever published the structure of a human endogenous retrovirus. Okay? And this is a virus that hides within our own DNA, ours.
I didn't even know that was possible.
Yeah. This is crazy, dude. 8% of our DNA is viral endogenous DNA. 8% of our genome, approximately 98,000 annotated insertions, are viruses that are like infiltrating, that have infiltrated. Dude, they're like sleeper cells, dude.
This is when people say the aliens created
No,
us. They would
yeah,
argue,
this
look,
is-
this is
it's-
exactly
yeah,
what I'm talking
yeah,
about.
no, like-
the little viruses are there writing our stuff.
It's crazy. It's crazy to think about. So first of all, I was like, how does this even get in? Why is this in my DNA, bro?
Right.
Like, what's going on? Okay, so it's crazy. These things are called retroviruses. A retrovirus is a virus that has, instead of using DNA as its genetic material, it uses RNA as its genetic material. And what it does is when it infects a cell, it uses something called reverse transcriptase. Usually transcriptase goes from DNA to RNA. This is the reverse process. So it's taking RNA and it's making double-stranded DNA. So that's reverse transcriptase. And then what ends up happening is that reverse transcriptase goes inside the nucleus of our cells.
Yes.
And then there's something called integrase, which is another viral protein that takes that DNA, that bit of viral DNA, and sticks it inside our chromosomal DNA. Now, if this happens to our skin cells, okay, we got like some viral DNA. But if it happens to our sperm and egg, now the next organism that gets that sperm and egg, all of the DNA
has it.
is going to have it, right? These are called germline mutations.
I was literally saying,
right?
it's kind of going back to our CRISPR story.
Yes.
It's the germline.
It's the germline mutations, right?
Yes.
So this is a germline insertion that happens. And then now the virus becomes something called a provirus, which is something that's just hanging out in our DNA.
Yes.
8% of our DNA
comes from this.
Dude, that's 1 in 12. That's like a lot.
That's a lot.
That's a lot.
I had no idea this was even a thing when
Right?
we first put the story in the show
this
notes.
is crazy.
I was like, this is fascinating.
Yeah. So all of that junk DNA, 8% of that is viruses just hanging out. Okay. Now, most of these viruses, they get mutations over the years and they can become inactive. Okay. But there's a specific virus called the human endogenous virus K. The HERV-K virus.
HERV-K.
It's a provirus now because it's within our DNA. But this thing is the most recent infiltrator. Okay, and it's because it's the most recent infiltrator, it has the fewest number of mutations, right? Which means it's still a problem. Okay, and it actually, so in most healthy cells, there's epigenetic silencing. Okay. Our DNA has figured out, I don't know who you are.
Yeah.
I'm gonna just put a bunch of like random stuff on you, so that no one ever transcribes you into RNA and then you become a protein. Okay,
Yes.
so our DNA is very good. If you're healthy, the cell is healthy. It's like, I don't know what this is.
It's like a bouncer at a club.
It's like
you don't
you're
look
already
21.
in here, but you're cut off.
Yeah.
You know, it's like, I don't know how you got in, but no more drinks. Okay. Every once in a while, you get something like a cancer cell, or you get autoimmune disorders. The cell is losing control, and this virus can start acting up. Okay, so it's actually been detected, this HERV-K envelope, the envelope protein that is the the protein that encodes for the envelope of the virus.
Yes.
That's the thing that's snuck in.
Yes.
Right? That has been known to be expressed in cancers like breast cancer, ovarian cancer, prostate cancer, melanoma, and also in autoimmune disorders like lupus, diabetes type 1, SLE, these kinds of autoimmune disorders. You can actually see the cells that are affected express this HERV-K protein.
Got it.
Which means that it is actually a problem.
Okay.
Right?
Got it. Yeah,
which
yeah.
means it needs to be solved.
Yes.
Okay?
Yes.
And in biology, like half the battle is solving the structure.
Right.
Because all of biology is lock and key, right? You solve the structure of the thing, then you can figure out what other structures can latch on and like mess with it and like inactivate it and things like that, right? So the first battle is to actually solve the structure.
That
Solving
makes
the
sense.
structure for this thing was really hard.
Okay.
Okay? Because it's this envelope protein of a, it used to be back in its heyday, right? Before it snuck in and now it's like just like an infiltrator. Back in its heyday, it was the envelope protein for a retrovirus, right? And these envelope proteins are incredibly finicky by design. You want your envelope protein to be finicky because imagine you're a virus, right? And you're getting to the outside of a cell that you want to infiltrate. You want the protein to first be able to infiltrate through the membrane, and then when it's in the membrane, change its conformation to open up, right? So you want this thing to have this sort of metastability, is what it's called, where you've got two states. There's this pre-fusion state, and then a post-fusion state,
yes.
right? Pre-fusion meaning like, how do I get in? Post-fusion, how do I...
You
do I
have the
get-
Trojan horse go to the gate in one state, and then when it's inside the gate, it's
You got to open up. Yeah, right? So you want this metastability.
Yes.
Now, that becomes a real problem when you're trying to get structure,
Yes.
right? Because you can imagine you're trying to image this thing, but this thing is in two different
Yes.
conformations.
Yes.
So like you don't know what you're looking at.
Yes.
And it's just annoying.
Yes.
Right? So
Yes.
that's the big thing that this group solved. Okay? What they did, the first thing they did was they took the protein itself and they put in they tweaked the protein in a certain way that they could, like, they created these things called disulfide bridges, which are basically like staples on the protein. What you do is you put in cysteine, which is this particular amino acid that has sulfur atoms, and those sulfur atoms can find each other and sort of bond.
Yes.
So you create a more stable protein.
Yes.
That is still very much like the original protein. It still preserves a lot of the structure,
Yes.
but it prevents the protein from doing this finicky stuff.
Yes.
And then what they do was they use a groundbreaking technology, the 2017 Nobel Prize in Chemistry, called cryo-electron microscopy. Okay? It used to be called blobology.
Blobology.
Yeah, from all the detractors, all the people who would make fun of these cryo-EM people, it was called blobology. Because back in the day, what you do is you get a protein and you like image it with electron microscope.
Yeah.
But because of this problem of like, the proteins move around and then they're not stable and all this other stuff, and you would get blobs, giant blobs, and you'd be like, it kind of looks like this. So all of the X-ray crystallographers and the NMR spectroscopy people, they're like, look at these blobologists, right? Because with X-ray crystallography and with NMR, I can get down to atomic resolution. I can get to 2 or 3 angstroms of resolution, where I'm like, that atom is there and it's there.
Yeah, right.
The problem with X-ray crystallography and NMR is it's really good for soluble proteins, so proteins that are found, within the cell, doing their thing. But for these transmembrane proteins, it's very hard to crystallize. It's very hard to make them behave properly. Cryo-EM was a technique that probably could have been used for that. Along came the computing revolution. And now all of a sudden what you could do is you could take thousands of images in cryo-EM. What you do is you have a bunch of proteins that are sort of frozen in this state, using this thing called vitreous freezing, where you're not like freezing it with ice, because if you freeze it with ice, the protein's gonna like misfold, right? So you do this like weird, like sort of vitreous freezing where the protein retains its structure. You image it a bunch of times, and then you feed that thing to a computer. What you're getting is 2D shadows, right? The proteins are in different orientations. You get a bunch of 2D shadows, but a computer now can take all of those 2D shadows and say, what is the 3D thing that would create all of these 2D shadows? right? In 2012 was the big one when it came out, and there was finally a protein that cryo-EM had solved that no one else could solve with any other technique. And they solved it to 3 angstrom precision. And everyone was like, oh, it's here.
Yeah.
And then in 2017, they won the Nobel Prize in Chemistry. It was 3 guys, Joachim Frank, Jacques Dubochet, and Richard Henderson. So blobology now became like a Nobel Prize winning technology.
Ain't no bunch of blobs now,
Yeah,
huh?
exactly.
Where's your Nobel, huh?
I think they were very vindicated when they actually got it right. So these guys, they actually used the cryo-electron microscopy to get the data from this after having messed with the protein to create these staples and make it sort of stable, right? And this was a very big deal because now you've got the 3D structure for this thing. You can compare it with other retroviruses that have been solved, like HIV, like SIV.
Yes.
And this one is very different because the protein that they came out is like taller and skinnier than the HIV protein, which is interesting in its own right because now we've got this difference. We can now start creating antibodies to actually target this thing.
This lock and key thing you were just talking
Exactly. And that's what they did. They weren't done. They created monoclonal antibodies to actually target this thing. And what they could do now is you can actually have diagnostics where, you know, in autoimmune disorders, for example, right? You have these neutrophils, which are these kinds of immune cells. And what they do is they're expressed in these cells, and then in an autoimmune disorder, now your immune cells think that, oh, this is a bad cell, I'm going to attack my own cell, right?
Yeah.
So maybe now that we know the structure,
Right.
we can actually like prevent that from happening, right? We can train the immune system or maybe cloud these antibodies in such a way that the immune system no longer attacks its own. On the other hand, we can also have ways to early diagnose certain cancers because cancerous cells express this protein specifically, right? So if we get like a blood sample from somebody, we can have an early diagnostic where you have these monoclonal antibodies that'll go and actually attach to these cancerous cells, and you'll have a marker now. Be like, I see this HERV-K protein. That shouldn't be expressed. In a healthy cell, the epigenetics should totally silence it.
And it's not.
But the bouncer is not working, right? So
right.
now I can start therapies earlier.
They're still in the club getting too drunk. They haven't been cut off.
Yeah.
So let's go ahead
and try to figure out how to get these guys out.
That's really
right. It's the first time that we've done this,
and the idea is now that we've done it once, by confirming that this methodology is a means by which you can do so. This can likely be extended to other ones. This is the most recent, and all these.
And this is the one that's like most prevalent. But the idea of like sort of changing a protein's shape using these editing mechanisms, where now I can have this like stapled disulfide bridge that sort of stabilizes it such that I can now do cryo-EM. This is a pretty interesting technique. The same group actually had done it for other proteins, and then they tackled this one because this one's hard. This one's like,
if you can do it with this one,
then it's like okay, now it's like
now we can. Fascinating. This
it's a cool one.
I always love our like microbiology, immunology stories because people don't understand how well we can look at and analyze and understand things that are really small.
Like we know exactly where to put in the cysteine so that the sulfur staples to another sulfur, but it doesn't really change the rest of the structure. That's like to have like this thousands of atoms worth of protein and know exactly where to put this thing so that I'm just going to clip this part. Because that's the part that's like getting messed up.
I would like our next Department of Health and Human Services secretary to be able to describe in detail any number of these concepts. Because that is usually a good prerequisite to say you're going to actually understand the import of medicine for public health.
Exactly, mate.
But that's a good one. I like that one. And we've touched on CRISPR a lot on this podcast. We've touched on a variety, and it's interesting. Again, all of these innovations are happening simultaneously in these cross-domain areas that have not only independent value but also like collective value. As we move into what we're seeing is going to be this like biotech revolution.
It is. It really is. We're here
for a whole AI applying the tools, the fundamental hardware tools the creativity of
Yeah.
how we're problem solving.
Yeah.